Hello, everyone. My name is Alexander Fernandes, and this presentation is part of the IEEE International Conference on Flexible and Printable Sensors and Systems. This is part of the session Sensor Device Architectures and Smart Systems. So to get started, this presentation is Flexible and Wearable Ultrasonic Sensors and Method for Classifying Individual Finger Flexions, presented at Carleton University. So to start, the current method for the prosthesis control approach is to use surface electromyography as the main sensor strategy. In academia, multi-electrode arrays with a pattern recognition algorithm is used to control prosthesis limbs, but industry typically uses fewer electrodes with predetermined number of hand gestures to select from. The problems with surface electromyography in itself is that the electromyography sensors exhibit crosstalk and averaging between the sensors themselves, resulting in limited specificity in some of the deeper muscle layer activity that's occurring within the forearm region. So researchers thought, why not use ultrasound? And Sikdar et al. In 2014 used B-mode ultrasound to spatially resolve the muscle activity occurring deeper in the forearm. And they were able to obtain a classification accuracy of 98% when discriminating between the thumb, index, middle, and ring finger, 4 fingers. While Georgia Tech Institute then took the ultrasound linear array probe and connected it with a prosthesis device, and they were able to get the amputee to play piano keys with their fingers individually, with individual control over the finger movements. But using a linear array probe is bulky, so researchers thought, why not use a more ergonomic approach? So again, et al. Used 4 PVDF ultrasonic transducers and wrapped them around the forearm as shown in the figure to the left, and obtained a 98% classification accuracy when discriminating between 11 different hand gestures. This research is very good for, and what they came up with is very good for American Sign Language, but because they only researched on classifying between discrete hand positions, they didn't necessarily test for the individual finger flexions themselves. So the research has yet to be proved for prosthesis research in itself. So our goal was to use wearable and flexible ultrasonic transducers to continuously classify the individual finger flexions themselves. That way, our goal in the future would be to operate the prosthesis upper limb devices and other human machine interfaces with individual finger flexion control. So we designed 3 wearable ultrasonic sensors constructed out of 110 micrometer thick polyvinyl iodine difluoride film with an active area constructed to be 20 by 20 millimeters with silver coating, resulting in a total thickness of the sensors to be 280 micrometers. The measured ultrasonic frequency for the 3 sensors came to be 5.1 megahertz from the echo, the pulse signal. For our experimental setup, we chose to place the 3 sensors as shown in the left figure, 5 centimeters from the wrist on the anterior side of the forearm to measure the flexor muscles of the forearm as shown. The wearable ultrasonic sensors were connected to their own pulsar receiver, and the signal generator produced a square wave at a 1 kilohertz frequency to generate a frame rate of 1,000 frames per second for the M-mode ultrasound data recorded. The waveform digitizer hooked up to the PC had the sampling rate set to a maximum of 125 megahertz, and this resulted in a depth resolution to be 6.16 micrometers per sample. For the experimental procedure, we continuously flexed each of the individual fingers for 10 trial recordings for each finger. Each of the recordings lasted 6 seconds with 5 flexions on each of the fingers, resulting in 5 minutes of total recording for 10 trials for each of the 5 fingers, or 50 trials. Here, if we look at one of the trials that are recorded as an M-mode visualization, we can see the index finger flexion recording over 6-second period. Now, we can see here, this is an example of tissue motion activity occurring at varying depth regions in the 3 transducers themselves. And if we look in more detail describing what's happening, we can see that the index finger is going between an extended and flexed position, shown in the first wearable ultrasonic sensor, with 5 flexions occurring throughout the 6 seconds. For each of the RF signals recorded, what we did was we cropped out the region of interest that related to activity that we mostly cared about. Because in the shallow regions, there would be a large pulse due to the transducer effect. And at some of the deeper regions, there wasn't a lot of activity occurring due to that's the region of where the bone is, as well as some of the signal became too attenuated and not a lot was occurring at those regions. So we effectively got a 7.4 millimeter region of interest depth for each of the sensors. Overall, the signal analysis procedure to eventually classify the finger flexions themselves was first, the recorded data was pre-processed using the discrete wavelet transform. Then spatial and temporal features were extracted using mean absolute values, the spatial features, and difference between frames as the temporal features. Then 2 classifiers commonly used in surface electromyography studies and ultrasound studies was used using linear discriminant analysis as one classifier and multi-layer perceptron as the other classifier. Our performance evaluation, we used a 10-fold cross-validation accuracy, and we compared the classification accuracy and F1 scores between these 2 classifiers. So for the pre-processing, the discrete wavelet transform, we obtained the first 6 levels of the RF signal. And here, if you look at the right figure of the frequency spectrum, the blue waveform is the Fourier transform of the RF signal before the discrete wavelet transform is applied. And what we can see is that the majority of the signal power lies in the levels 4 through 6. So for this step, we pre-processed and obtained the detail coefficients and discarded the level 1, 2, and 3 detail coefficients because there wasn't a lot of information occurring at these frequency ranges. Then we obtained spatial features by using the mean absolute value between the depth samples within the wearable ultrasonic sensors. And the mean absolute value would be only occur along the detail coefficients of the same depth and level region, so that the temporal features would then be the difference between each mean absolute value at that given depth and level for the wearable ultrasound sensor channel, and subtracted from the same mean absolute value, but of the previous frame. These features that we extracted were then put into the linear discriminant analysis classifier and multi-layer perceptron classifier. LDA has been proven to be a good dimensionality reduction technique, and a multi-layer perceptron, we designed it with a 1 hidden layer and 15 nodes to keep it at a simple architecture. For the data, the sample data used to predict on each of the, by the classifiers, 10 frames were arranged into a single sample. And this means that a prediction would be made every 100 milliseconds in time for the finger flexion. This is to satisfy the potential use of a real-time system predicting which fingers currently being flexed every 100 milliseconds in time. And the classifier would choose from one of the 5 fingers, and the training and test data was split based on the trial number, effectively a 10-fold cross-validation based on which trial number would be used as the test data. Overall, if we look at the results, the multi-layer perceptron performed better than linear discriminant analysis. And if we look at the classification accuracy, the multi-layer perceptron had 92% accuracy, 92.5% accuracy, and linear discriminant analysis had 86.6% accuracy. In fact, the multi-layer perceptron outperformed the LDA in each of the finger F1 scores, except for the middle finger. Our thoughts on why this might happen could be because the middle finger has flexor muscles attached to both the index and ring finger, meaning that these 3 fingers share the same muscle flexors to perform the actual finger flexion motion. And linear discriminant analysis inherently tries to maximize the between class scatter and minimize the within class scatter for each of the individual finger flexions, whereas the multi-layer perceptron tries to just maximize the general overall accuracy. In conclusion, both of these classifiers resulted in F1 scores above 80%, showing that this would be showing that this accuracy is very high with the given preliminary study that we've just investigated. If we look, the multi-layer perceptron had an F1 score ranging between 85 and 99%, with the middle finger being the lowest, and the LDA F1 score was 82 to 88%, with middle finger being highest. In the future, more subjects will be recruited to verify this preliminary study, and we will include a rest position to classify what happens if we want to predict if the user's hand is not moving in a rest state. And finally, we would look into determining an optimal number of these wearable ultrasonic sensors and placement, such that for this study, we only looked at the flexor muscles as a preliminary study, but we will also look around the full forearm to include the extensor muscles on the other side of the forearm. Thank you for attending my presentation. I hope you enjoy. Thank you.